First off, I just want to say I've really enjoyed this meeting, and it's taught me how little I know about drought physiology, and I think I need to have one of my students take the course next time it's offered. I'm really in that genomics part of the bubble, but I think that what we've learned by employing modern functional genomic approaches to this problem should have ramifications throughout a lot of areas. So what I'm going to do today is tell you really the story that involves my lab of how we identified a new family of ABA receptors. And that was the key to this whole discovery from my lab, was a small molecule that came from that screen that started at the University of Toronto. So part of my talk is going to be just telling you about this side of the equation, which is really, that doesn't have much directly to do with drought physiology. And then I'll talk about our first steps towards translating the new knowledge that we've acquired to maybe making some improvements in drought physiology, focusing mostly or initially on experiments in Arabidopsis. And what we're trying to do is develop chemical tools and genetic tools for activating the ABA signaling pathway so that we can elicit some of the protective effects it has on stress tolerance. And some of the data I'm going to show today comes from collaborators who are structural biologists. My lab does not do any structural biology. In particular, Brian Volkwin's lab at the Medical College of Wisconsin has been a very close collaborator, as has Eric Shu at the Van Andel Research Institute. And then I will be showing some unpublished data from two postdocs in my lab, Masanori Okamoto and Asaf Maskuna. And I should also just point out that I'm going to tell you the story of how my lab came to this new receptor family, and we found the founding member, which I'll tell you about, named Pier1. But a lot of other labs came to this around the same time, including Erwin Grill's lab that has given this gene family I'll tell you about another name. They call it the ARCAR family. Okay, so for good reason, Arabidopsis geneticists and biochemists and investigators in a lot of systems have been looking for components involved in ABA signal transduction for quite a long time. The experiments I'm most familiar with originated in Arabidopsis, starting with genetic screens that Martin Korneef did in the 1980s. And that identified a number of components, some of the downstream components, but nothing ever resembling a receptor came out of those screens. And the take-home message on this part of the talk is that the reason those receptors never emerged from the genetic screens is probably because of the extensive sequence redundancy that exists in the Arabidopsis genome, and that often translates into extensive functional redundancy. And this is just a little cartoon illustrating the segments of the Arabidopsis genome that shared a common ancestor and are derived from some ancestral duplication. It's not just a quirk of Arabidopsis. Whole genome duplications seem to be quite pervasive throughout plants. And this creates a general problem for geneticists who try to identify genes by their loss of function phenotypes. If you happen to be working in a system that has multiple copies of a gene involved in a process, it may end up being invisible to the reach of forward genetics. And that is indeed what we think happened in the case with ABA receptors. As I'll tell you in more details later on, it looks like the ABA receptors are encoded by a 14-member gene family. And to date, the loss of function phenotypes for about 10 of those family members that have been characterized don't seem to have any real noticeable defects in ABA perception. And that's because if you knock out one of the receptors that perceives ABA, there's several other that can compensate for its loss. This is the classic problem of redundancy and is due in part to all of the genome duplications that have generated these duplicate copies. And the chemical approach that was so unpromising initially to John, that worked out quite nicely for us, led us to a molecule that allowed us to break down this redundancy. So we were screening through the libraries that John mentioned, and I won't go into too much detail about those screens, But we found a molecule that looked very much like it was activating the ABA signaling pathway, and I'll show you that data momentarily. But we could also tell that it wasn't like ABA in all of its effects. It had certain selectivity and was really only acting as a good ABA agonist or activator in seed tissue. And so when we used this molecule for a genetic screen, we didn't have all the other receptors competing with it because of functional redundancy. And so this simple molecule allowed us to identify the gene PIR1 that I'll tell you about, and it essentially allowed us to bypass this functional redundancy problem. And I think that this is one of the more compelling reasons to be taking this small molecule approach for dissecting pathways in plants. So I'm going to talk about two molecules today. One of them is quite selective in its effects. That's pyrabactin, and I'll have a lot to say about it. And then I'll tell you about a new molecule that's structurally related to pyrabactin that activates slightly more receptors and is able to activate the vegetative ABA response pathway. So without going into all of the technical details, the long and the short of it is that we used chemical libraries that are used by people in ag chemical discovery or in drug discovery for mammals. And we looked through them to find compounds that affected Arabidopsis seed germination and hypocotyl phenotypes. And we screened many thousands of compounds and found a lot of hits, but one of the ones that jumped out at us from the very beginning was this molecule we call pyrabactin. And we call it pyrabactin because it has this pyridyl ring that's critical for its bioactivity. So this analog is, as far as we can tell, biologically inactive. The pyrabactin itself inhibits seed germination. As I'll show you, that's because it's activating the ABA signaling pathway. And so the abactin part is for ABA activation. And as a master's student, I had worked on ABA, and I was well aware that there was a lot of controversy surrounding the identity of ABA receptors. And I got interested in understanding how this molecule worked. And it's quite obvious to anyone who knows anything about seed physiology that there's lots of different ways to chemically block seeds from germinating. And so we did a number of experiments that I'm not going to go through that ruled out some of the most likely scenarios. And then we finally tested some mutants that were not sensitive to ABA. So these are two mutations that were isolated from those screens I described earlier for ABA insensitive mutants. And you can see that ABI1 and ABI4, these two ABA insensitive alleles, would germinate on our compound, which told us that we had to have a functional ABA signaling pathway for this molecule to do its magical job of inhibiting seed germination. We also examined mutants that are Arabidopsis mutants that are unable to make ABA or have greatly reduced levels of ABA due to defects in their biosynthetic enzymes. And those showed normal sensitivity to this compound. So that allowed us to conclude that it wasn't just that the molecule was triggering ABA biosynthesis, but that it was doing something that required the signaling pathway. We next did whole genome microarray experiments to try and compare the effects of our molecule to those of ABA, first looking in seeds. And so this is just a scatter plot of the fold change. In each case, it's with respect to a control. And you can see there are a lot of genes that respond positively and negatively to ABA. And what we could see was that a gene that responds positively to ABA also responds positively to our molecule pyrabactin. And we had a very nice positive correlation here. And more importantly, we weren't seeing a lot of other transcripts being induced by pyrabactin. So that told us it wasn't activating many other pathways, at least at this transcriptional level. So based on this experiment, we felt pretty confident that we had a molecule that was talking to something central in the ABA signaling pathway, at least in seeds. When we did the same experiments in seedlings, so about 1 week after they've germinated, where really a lot of, where we could measure a lot of the responses that would be interesting to drought physiologists, or at least start measuring those, we didn't see this nice correlation between ABA effects and pyrabactin. There is a slight positive correlation. With the significance filters we used, there were about 50 pyrabactin responsive genes that were also ABA responsive, but by and large, the 1,000 or so genes that responded to ABA were not responding to pyrabactin with a significance cutoff. So this is the key piece of data that told us that pyrabactin, although it's like ABA in seeds, is quite different in vegetative tissue or seedling tissue after the seeds germinate. And that told us that there was some selectivity to this molecule in its interaction with the ABA signaling pathway. The physiological correlates of this are, and this is where my limitations as a drought physiologist will become obvious. This is one of our assays in the lab that we use for ABA effects on vegetative stress tolerance. It's just a water deprivation experiment. At the end of the experiment, which is about 3 weeks, we re-water the pots and then look at survival. And in these experiments, ABA always gives us very nice, clean survival after this catastrophic drought treatment. And in these experimental conditions, pyrabactin is not able to do what ABA can do. And understanding the basis of that and trying to get a molecule that can do what ABA does in vegetative tissue became a subsequent interest in my lab. So given the unique properties of this molecule, we were very interested in understanding how it could turn on the ABA pathway. And I am trained as a geneticist, and when it comes to understanding mechanism of action, I tend to take the genetic approach. So what we did was we looked for Arabidopsis mutants that would germinate on pyrabactin. We found a number of those. Most of them mapped to the same locus, which we called pyrabactin resistance 1, or PIR1. And we used map-based cloning to identify what the molecular nature of the defect was in our PIR1 mutants. And I should point out that very early on, after isolating these pyrabactin-insensitive mutants or pyrabactin-resistant mutants, we could see that the PIR1 mutants had normal ABA sensitivity, which we now know is a consequence of redundancy with the other ABA receptors. At the time, we didn't know that, and we were a little worried that we were barking up some tree that might not have anything to do with ABA. So the map-based cloning told us that PIR1 was encoded a protein that was part of a large family of ligand-binding proteins that are fairly ancient. They can be found in bacterial and eukaryotic genomes. The domain that they share in common has been named the START domain by Aravind, a bioinformaticist at NCBI. The founding member of this family is called STAR-D, and it binds cholesterol and is involved in transport of cholesterol into the mitochondria as a rate-limiting step in steroid biosynthesis in mammals. Their plants have genes that contain this classic START domain, and quite a number of genes that also contain this anciently diverged START domain that's also called the PR10 or the BETV1 domain. And the structures for each of these representatives have been published long ago, and they share a common topology, which is 7 antiparallel beta sheets that surround this C-terminal helix, and in between there forms a ligand binding pocket. So the long and the short of it was that the map-based cloning told us that we had identified a ligand binding protein, putative ligand binding protein, and we had a ligand, and it was from there that we started thinking maybe this was directly involved in binding to our ligand and directly involved in ABA signaling somehow. But the challenge was really to figure out how. And I should say that in addition to identifying a number of point mutants throughout the coding sequence that changed a number of amino acids, including early stop codons, when we put the GFP fusion of PIR1 back into our mutant plants, it restored function, which these two lines of evidence together made us very clear we had identified the right gene. And you'll note that the GFP fusion protein looks basically like a classic soluble protein. It has a nucleus cytoplasmic distribution that's essentially indistinguishable from what you would see with just GFP on its own. So this does not look like it's a membrane localized signaling component. And we can talk about that later if anyone's interested. So the challenge then was to figure out how this novel ligand-binding protein connected our molecule, pyrabactin, with ABA signaling. And I have to say that what we did next was a little bit of a leap, and it was really inspired by some of the recent meaning the last 10 years, recent understanding of how a lot of plant signaling molecules and hormones work. And the paradigm example, the one that I think should be familiar to many of you, is that of auxin, or indole acetic acid. And IAA stabilizes a protein complex between an F box, an SCF containing F box, basically a ubiquitin ligase complex, and the OX-IA proteins, which are transcriptional repressors for the most part of auxin action. And once stabilized, this complex allows those to be ubiquitinated and degraded. And this was a really stunning discovery that came from Mark Estell's lab and Adeline Leiser's lab. And it showed that plant signaling molecule could stabilize a protein-protein interaction. And shortly thereafter, conceptually almost identical mechanistic example came from studying the mechanism of jasmonate signaling, where SCF CoI1 complex is stabilized with Jaz proteins. In the case of gibberellin, the GID1 receptor forms a complex with DELA proteins, although it's not directly involving an SCF complex like with jasmonate and auxin. So the long and the short of it was that we had this idea and this inspiration from prior hormone work that plant signaling molecules like to stabilize protein complexes. And so our wishful model at the time, and this was right around the time when I showed up at Riverside, was that PIR1 might interact with some downstream effector in the ABA signaling pathway, and it was that effector that talked to ABA signaling. And if it was working like the way other plant hormones work, this might be able to be identified by virtue of a ligand-controlled protein-protein interaction. At least that was sort of the wishful thinking at the time. And one of the reasons we pursued this line of logic was because it wasn't actually that hard to go fishing for such a protein-protein interaction using the yeast-2 hybrid experiment. So we used PIR1 as bait in a yeast-2 hybrid, and we added our chemical to the yeast growth medium, and we looked for clones or proteins that would bind to PIR1 in this yeast assay in the presence of our chemical, and then we retested them all. We only got 2 clones that came out of this screen, and both of them interacted with PIR1 in a manner that depended on the presence of our compound. That dead analog that I showed earlier wouldn't promote the protein-protein interaction, and there was no interaction in the absence of the compound. And I should say that we've done quite a lot of biochemical work to show that this isn't just a yeast artifact. PIR1 and the target protein we identified physically interact in vitro as well in a manner that is stimulated or promoted by our ligand. But the real exciting day was when we tested ABA shortly after identifying these clones, and we could see that ABA also promoted this protein-protein interaction in yeast, which was really the first piece of evidence we had that connected PIR1 and pyrabactin to a meaningful ABA response, which was a great sigh of relief. The next connection to the ABA signaling pathway that was also very gratifying was that the clone that we pulled out of this yeast-2 hybrid screen was a relative of ABI1 called HAB1. HAB1 and ABI1 are members of a subfamily of protein phosphatases called the PP2Cs, and a lot of genetic evidence that was published long before we identified PIR1 had connected this phosphatase gene family to ABA signaling. And the role of the phosphatases from the genetic evidence was that they negatively regulate signaling. The mechanism by which they negatively regulated signaling was not clear, but it was clear that if you made loss of function mutations, and in particular multiple loss of function mutations in these phosphatases, one would see constitutive activation of ABA signaling and ABA hypersensitivity, which told the geneticists who did that work that they negatively regulate ABA signaling. And so the way we envisioned then that our protein could regulate ABA signaling was that if it inhibited this inhibitor, that would turn this double negative into positive regulation of ABA signaling. Fortunately, that was pretty easy to test because these are both soluble proteins. The phosphatases had been well characterized in vitro, and so we expressed recombinant phosphatase and included in these experiments either wild-type pyrA1 or one of the point mutants that came from our genetic screens, and then looked at the effects of ABA in the presence of these proteins on phosphatase activity. In the absence of ABA, phosphatase activity is essentially indistinguishable from control reactions lacking the pyrA1. And as we increase ABA concentrations, we inhibit phosphatase activity, and that's greatly diminished in this point mutant, which we now know affects the interaction between the receptor and the phosphatase. So what this told us was that the function of ABA, at least in this particular system, is to inhibit phosphatase activity, and that inhibition is dependent on pyrA1. Working in Arabidopsis, there's very high standards in terms of the genetic proof that one needs to support a model. And also, our work came on the heels of many other claims of ABA receptors, one of which was retracted, some of which are still controversial. And we knew that we were going to have strong genetic evidence to support our model. And so, as I mentioned, the single pyrA1 loss of function mutants that we isolated didn't have any ABA phenotypes, so we constructed a quadruple mutant that removed pyrA1, PIL1, PIL2, and PIL4. Those were the three closest relatives of pyrA1 that had high expression levels in public microarray databases. And in that quadruple mutant, we could see that ABA responses were affected in a number of different measures. One is root length growth. We're not showing the control here, but the Columbia roots are inhibited by ABA. The quadruple mutants are greatly insensitive to that inhibition. This is one of several ABA responsive genes we've looked at. In response to ABA in Columbia and Landsberg, we get nice induction of this particular marker as measured by qRPCR. And the induction is reduced but not abolished in our quadruple mutant. And really the next, I would say, breakthrough in this came from an experiment that was done in collaboration with my former neighbor and collaborator, Jinkong Zhu. And his lab studies a group of kinases that Shinazaki's lab and Sally Aspin's lab had identified as being activated by ABA in a number of different systems. And so they go by different names, but in Arabidopsis, they're now known as the SNRK2 kinases. And this is an in-gel kinase assay in which a fragment of one of the AREBs or the ABFs, these are transcription factors that are necessary for binding ABREs and are involved in ABA signaling. This is measuring the phosphorylation of a fragment of one of those transcription factors in an in-gel kinase assay where Arabidopsis is pre-treated with ABA or not, and then you run a gel, you renature it, and you look to see what in that can phosphorylate the transcription factor. And in wild-type controls, you always see these two bands, which corresponds to 3 of the 10 SNRK2 kinases in Arabidopsis. And the key point here was that Hirofuji and Jinkong Zhu showed that our quadruple mutant is greatly debilitated in its ability to activate this cluster of kinase activities in the in-gel assay. And so from that, we proposed a model that goes for what our view of core ABA signaling is. And it goes something like this. In the absence of ABA, the protein phosphatases like ABI1, HAB1, the clade A PP2Cs are active, and they can dephosphorylate the SNRK2s. A lot of biochemical work from Shinazaki's lab and others had shown that the SNRK2s have to be phosphorylated on an activation loop to be activated. And that in the absence of that phosphorylation, they're not able to phosphorylate downstream factors like the AREBs, or AREB1 in particular. When ABA comes along, it binds to the receptors, and I didn't show the data, but we had binding data, which I'll come back to in a moment. And that promotes the receptors to bind to the phosphatases and inhibit their activity. In vitro, the SNRK2 kinases can autophosphorylate on that activation loop, which is necessary for their activity. It's not clear if autophosphorylation is sufficient for activation in the pathway, or if there's some other kinase in vivo that's phosphorylating them. But once they are relieved from this inhibitory dephosphorylation activity, they become activated somehow, which allows them to phosphorylate downstream target factors, such as the AREB transcription factors. And I should say, Jin Kong, I had played a minor role in this work, but Jin Kong took one member of each of these, so one of the receptors, PIR1, one of the phosphatases, one of the SNRK2s, and one of the downstream transcription factors, and he showed that he could reconstitute ABA-mediated phosphorylation of that transcription factor with just those 4 components in a test tube. And another sort of key piece of genetic evidence that, again, this comes from Shinizaki's lab and Jin Kong's lab, is that a triple mutant that removes those 3 SNRK2 kinases that are activated by ABA is essentially dead for ABA perception. So both Shinizaki's lab and Jin Kong Zhu's lab have shown that if you remove the 3 critical SNRK2s involved in ABA signaling, that essentially inactivates all ABA perception. So it's the combination of the genetic evidence and the biochemical evidence that has led to the idea that this is the core signaling pathway with these PIR-PIL RCAR proteins sitting at the top and controlling phosphatase activity. And this is a very simplified and pared down version of the pathway. I should point out that in the last couple years, a number of papers have come out complicating or enriching the picture of this pathway to show that it branches at the SNRKs. In addition to the transcription factors that are phosphorylated by the SNRKs, there's also a critical anion channel involved in guard cell physiology called SLAC1 that can be phosphorylated by the SNRK2s, and some evidence that a potassium channel may be inactivated by the SNRK2s. And Julian Schroeder's lab recently published a paper that may start to integrate calcium-dependent protein kinases into this pathway. So the long and the short of it is that we think this is based on the genetic and the biochemical evidence, that this is the bones of a basic signaling pathway that branches at the SNRKs to control the various outputs that ABA controls. And obviously, the future is going to be connecting or not the other known factors in ABA signaling to this pathway and figuring out how they work. Okay, so we collaborated with some structural biologists, and the real question after this work was published was, how do the receptors inhibit phosphatase activity? And there were like 5 different groups that were involved. I collaborated with 3 of them, and one of them in particular I'm going to show. This is data from Karsten Melcher and Eric Shu, and they caught the receptor PIL2 in its 3 states, which we think are critical for its activity. That is, on its own, complex with ABA, and then in a ternary complex with the phosphatase. The PIR1, and this is PIL2, PIL2 and other receptors that have been, for which a crystal structure is now available, look like pretty much every other start protein for which there's a crystal structure. They have 7 antiparallel beta sheets that enclose this C-terminal helix, and there's a central ligand binding pocket that's surrounded by a number of loops. One of the loops in particular is quite important, and it's been given the name the gate for reasons that'll become obvious in a moment. And embedded in the middle of that loop is a serine, although it's not totally clear in this alignment here. That serine is invariant throughout the receptor family. These are all of the Arabidopsis receptors, and this is a Fiscometrella ortholog and a Maze ortholog of PIR1, or at least the closest matches in blast searches. And so that serine is present basically across all the receptors. And when ABA binds, if you go back and forth, I think it's pretty obvious, what it's doing is it's stabilizing via contacts with residues in the gate, this alternate conformation of the gating loop. And that causes that serine, which starts off pointing into the pocket, to point out, that could pop out. And the normal substrates of the phosphatases would be phosphoserine or phosphothreonine. In the ternary complex, what is obvious is that serine is docked into the active site of the phosphatase. Those are its active site magnesium ions. There's a water connect that directly hydrogen bonds between the serine and these one of the metal ions. So ABA converts PIL2 into an inhibitor of the phosphatase by allowing it to dock into the active site and block access substrates. So essentially, this looks like a competitive inhibition mechanism. Okay, so one of the surprises, though, was that although almost all of the contacts between ABA and the receptor occur inside the pocket, there was one water-mediated contact between a residue on the phosphatase and the ketone, the ring ketone of ABA, and this residue was called the lock. And it's necessary for stabilizing the complex that forms. If you mutate this tryptophan to an alanine, the receptor is unable to inhibit phosphatase activity. That tryptophan residue is present on a loop that is highly conserved within the clade of phosphatases that are inhibited by the receptors. So not every PP2C in the genome has this tryptophan. It's just the ones that are regulated by ABA, with one exception that appears to be not regulated by the receptors. Okay, so just to summarize then what the structures told us, the ABA receptors are allosteric switches, and they have, in very simplistic terms, they have an off conformation and an on conformation. The on conformation is with the gate closed, and that allows the receptors to dock into the phosphatases and inhibit phosphatase activity. So in simple sort of Pac-Man or pizza cartoon here, there's two states, and I'm just showing the gate here to be open or closed with ligand bound. And what I'm going to talk about for the rest of the talk are our attempts to identify basically tools for turning these receptors on, either chemically with new ABA agonists or genetically with constitutively active receptors. And this, although the context I'm spinning these goals in for this talk is for drought tolerance, we also need these tools for understanding the functions of the genes. Because of this tremendous redundancy, we are having challenges to understand the functions of defined receptors. And with tools that allow us to selectively activate one receptor or another, we hope we'll be able to better dissect the roles of the receptors. But obviously, if we have a good agonist that turns on enough of the receptors, we think we should be able to induce the ABA pathway and whatever benefits that carries with it for stress tolerance. Okay, so I mentioned already that pyrabactin, although it was great for this seed response and identifying PIR1, it doesn't do much in the vegetative pathway. And one of our first questions really is, what do we need, which receptors do we need to activate to turn on the ABA signaling pathway outside of seeds, so in vegetative tissues after seedling establishment. And one of the sort of worst case scenarios that we had initially was that we might need an agonist that's basically mimicking ABA in every respect, that activates all of the receptors. That might be what you need to turn on this pathway. Alternatively, it may be that a small number of the receptors play a particularly important role in the vegetative responses. In terms of why pyrabactin is so selective for seeds, we've published a little bit on this in terms of the structural basis for it, so I won't belabor the point. But what we think in the case of pyrabactin, we think part of the reason that the molecule is so selective in its effects on seeds is that its receptor, its primary receptor, the one that we know genetically is critical for activity, is really highly expressed in seeds. Pyrabactin also activates this receptor PIL5, although it's more of a partial agonist for that receptor, and that didn't come up in our genetic screens. So we think that its primary target happens to be the most abundantly expressed transcript in seeds, and that it's the intersection of the chemical's affinity for that particular receptor and the receptor's expression pattern that generates this selectivity. Okay, so to try and figure out how we could get past or get other agonists that might activate more receptors, we just did another brute force screen, but this time incorporating some of the knowledge that we've gained about the pathway and some of the tools that we've developed. And so I've already introduced that we can measure the interaction of the receptor and the phosphatase in the yeast assay. And so we took our yeast strains that report activation of the receptors in yeast and screened through the same chemical libraries, or actually not the exact same ones, but conceptually the same kinds of libraries that we already screened through, and we looked for compounds that would just activate more receptors. We actually found some new agonists that are selective for other receptors, but I'm just going to focus on this new molecule we've identified called quinabactin, and you can see in the yeast experiments it was able to activate many more receptors than pyrabactin is able to. The yeast strains are really not the best measure of an agonist's activity because they're very sensitive. But this is at least the screening trick we use to identify this new molecule. And you can see that, like pyrabactin, it's a sulfonamide, and that sulfonamide linkage turns out to be critical to the interaction with the receptor's binding pocket. So unlike pyrabactin, the next few slides are really just to show you that this molecule is better than pyrabactin, at least from the perspective of activating the pathway more broadly. So this is a MAP kinase, MAPKKK18 GUS reporter line. It's just one of the reporter lines we use in the lab to measure ABA activation in a whole sort of seedling assay. And you can see pyrabactin does nothing to these GUS lines. ABA activates the marker as expected in pretty much everywhere with a higher level in guard cells. And our new molecule, quinabactin, gives us comparable levels of marker induction. These are 25 micromolar, both of all compounds tested. Most importantly, while pyrabactin is not able to give us that nice positive correlation we saw in seed tissues, when we profile the effects of ABA and quinabactin in seedling tissue, we get back that nice positive correlation that we could see in the seeds. And again, there's not a lot of, quinabactin's not turning on a lot of genes that ABA doesn't itself turn on. We've used the in vitro phosphatase assays to measure the potency with which different compounds activate the receptors. We can assay all of the receptors with the exception of these three in gray, which for some reason do not express well in E. coli, or at least don't come out of E. coli in active forms. And as expected, I didn't explain this, but some protoplast experiments had shown that with the exception of pill 13, the remainder are sufficient for ABA responses in a protoplast assay, and we see that ABA is able to activate all these receptors in vitro assays. With an IC50 value for pyrrh1, it's around 200 nanomolar. Pyrabactin, as I've already mentioned, prefers pyrrh1 into a lesser degree pill 5, and it's much less potent than ABA, about 4-fold less potent. And quinabactin is able to activate 5 of the receptors, and on pyrrh1, it's about twice as potent as ABA is. That's unusual. For most of the other receptors, its potency is comparable to ABA. So in terms of the physiological consequences here, it's not just gene expression. When we spray wild-type plants with quinabactin, we're able to cause guard cell closure. That doesn't happen with pyrabactin. Guard cell closure will occur in a mutant that's unable to make ABA, but it can't occur in an ABA-insensitive mutant, the same mutant that we used earlier. So this tells us that quinabactin's effects on guard cells depend on there being a functional ABA signaling pathway there. That's just fully consistent with the way we think this is acting, which is to activate these 5 receptors. This again illustrates the lack of sophistication we have in drought physiology, but I think it illustrates the main point. So these are water deprivation experiments where we, after withholding water, we spray every 3 days with either ABA, pyrabactin or quinabactin, and then measure wilting. And ABA, not surprisingly, delays wilting in these experiments, as does quinabactin. The inset numbers are for the survival assays we've done. Quinabactin can also promote that survival in the after water deprivation, just like ABA does. Okay, and its effects are not just restricted to Arabidopsis. We've looked at soybean, barley, a number of other species, and this is just that same drought experiment done in soybean, showing that both quinabactin and ABA can delay water loss and delay wilting under water deprivation. Okay, so just to summarize then, what we've learned from identifying this new agonist is what we need to do to turn on this vegetative pathway. Before, we could only activate the seed pathway, but not the vegetative pathway. Now we're getting good activation of both pathways, and we don't need to activate every receptor to do that. And that's going to be very important going forward as we try to design and identify new agonists that elicit stress tolerance. What we're doing now is combining this tool with loss of function genetics to try and more specifically map out if all 5 of these receptors need to be activated, or if it's just a particular subset that's sufficient to elicit all of the ABA responses we're measuring. Okay, so my collaborator Brian Volkman has determined a crystal structure of quinabactin bound to PIL2, one of the receptors it activates. And I'm just going to go over this briefly. So the question is, from a structural level, why is this new molecule so much better than the first one we identified, pyrabactin? And I mentioned before that there was this contact between a tryptophan on the phosphatase that makes a water-mediated contact to the ring ketone of ABA, and that's called the lock residue. And pyrabactin does not have a hydrogen bond acceptor there to interact in that way. And some of the structural data that I didn't show has shown that pyrabactin can actually rotate in the pockets. It can bind to the pockets of some of the receptors it doesn't activate and bind in a non-productive orientation. Quinabactin, on the other hand, doesn't do that. It seems to bind to the pockets of several of these receptors. And we think probably a key reason to why that's occurring is because it has a hydrogen bond acceptor that allows it to interact with that tryptophan lock, which may stabilize its binding in the pocket. And that's something we're trying to nail down a little bit at the moment. Okay, so to summarize then, this new agonist is more potent than pyrabactin. It's a little more potent than ABA in some receptors. It's shown us what's sufficient for which receptors are, sufficient to activate for getting drought tolerance. And it's informed us that we don't need a pan-agonist that activates every single receptor for that effect. And we're now trying to optimize this molecule and try and make it a better tool for activating stress tolerance and the ABA pathway. Okay, so the last part of my talk will be on using genetic approaches to essentially reach the same goal. And this is by trying to engineer constitutively active receptors. The basic idea was that what ABA does is it stabilizes this altered conformation of the receptors. And we would like to try and identify mutations that mimic that on conformation or closed conformation. And we reasoned that there might be a couple classes of mutations that could get us there. We imagined that perhaps a mutation, the right mutation, we didn't know which one, but the right mutation in a pocket-located residue might cause that gate to close. Or perhaps a mutation at the interface with the phosphatase could stabilize the phosphatase interaction and promote the on state. So what we did was we took all of the residues that were either involved with ligand binding or at the phosphatase interface, and in some cases there are residues that are involved in both, and we made every possible mutation at those sites using some PCR tricks, and then used our yeast strains to measure whether or not the specific mutants we created were active or not. And this is a fairly large effort led by a postdoc I already mentioned named Asaf Moscona. we're using these mutants for a lot of other purposes in the lab as well. And what we found was that of the 39 residues that we targeted, there were 10 residues in which we could identify activating mutations. And those are simply identified by, so this is our control here. In the absence of ABA, PIR1 will not interact with the phosphatase in the yeast strain. When we add ABA, we get nice blue yeast that's reporting the protein interaction. And then each of these mutations here are specific mutations in these residues that promote the receptor to interact with the phosphatase without added ABA. And I think what you can see from this figure is that they cluster together in the gate loop, which is perhaps not surprising, and then in the helix, the C-terminal helix that forms important contacts with the phosphatase. And then there was another outlier that's three-dimensionally very similar in space to that C-terminal helix that was another hotspot for these mutations. So when we characterized the biochemical effects of these single mutations, what we saw is that they weren't sufficient to fully activate the receptor on their own. They partially activate the receptor, so this is increasing amounts of receptor to phosphatase in the presence or absence of ABA. And not surprisingly, these are the wild-type controls in blue and red, and then the green is the specific mutant that we characterized from this screen. And if it were activating the receptor as good as ABA, it would overlie on the red line, and you can see none of them do that. So we started combining mutations from the different regions that we identified, the gate, the helix, and this outlier here. And what we found when we made triple and quadruple mutant combinations of our single mutants, we were able to construct receptors that were nearly indistinguishable from an ABA-saturated receptor. When we take those triple or quadruple mutations, they reside in highly conserved residues. And so what we're showing here is that we can just port those mutations into other receptors, and it allows them to become constitutively activated. And it doesn't matter which particular phosphatase pair we're measuring, which is a technical detail, but an important one. So this tells us that we know how to engineer lots of constitutively active receptors, and we have the tools to make these turn-specific receptors on in vivo genetically. This was published in PNAS last year, so I'll very briefly just say that when we introduced these transgenes, this is the PIL2 constitutively active receptor into Arabidopsis, we saw very hyperdormancy in the seeds, and a lot of ABA-regulated genes were upregulated in our constitutively active transgenic lines. For reasons that we still don't understand, the transgene silences, and it's not actually classical silencing, it's just that the protein's not being expressed in vegetative tissue. So our analyses were restricted to seeds, but we could see by a number of assays that this constitutively active receptor was activating ABA signaling in seeds. So we did the trick that, it's actually a trick that originated with Shinozaki. It was used for the CSPB that was used in the Monsanto drought tolerance work. We tried to express these constitutively active receptors under a drought-inducible promoter to see if we could get stress-inducible activation of the pathway, and more so than would occur in just a wild-type plant. And we did that for 2 receptors and compared it to either just expressing the wild-type receptor or the constitutively active version of the receptor. And this is just to show you that the constructs actually do lead to drought-inducible expression of the transgenes. But more interestingly, and we're just starting this work now, we're analyzing these transgenics for a number of stress-related phenotypes. This is a salt stress assay. Again, this is just Arabidopsis. This is after 2 weeks growth on 250 millimolar sodium chloride. That's enough to kill about half the wild-type controls. And in our CA lines, we get substantially less of the seedlings dying. We've done water deprivation experiments where we allow the plants to grow fine for about 6 weeks under short days, and then we stop watering, and we measure water loss over time. This is at the 2-week time point, but we can see that our CA lines are losing, our inducible CA lines are losing water more slowly than either the wild type receptor being overexpressed on that inducible construct or our wild type Columbia controls. So the first round evidence is that we're getting some promotion of stress tolerance with these inducible constructs, and it seems to be better than just inducibly expressing the wild type receptor. And this is true for multiple receptors that we've tested. Okay, so to summarize the constitutively active part, and this is really the end of the talk, we've figured out methods for engineering the receptors so that they're always on. We're using that for both these drought experiments, but also we're using them to try and dissect the roles of individual receptors in ABA signaling. The CA receptors work in vivo. They're sufficient to activate ABA signaling. And the first round evidence is that we're getting improved stress tolerance using these constructs in Arabidopsis. And just to summarize overall, so by identifying this new agonist, selective agonist, we were led to a new family of ABA receptors that seem to work through this core pathway. The receptors are allosteric switches that function to inhibit phosphatase by docking into their active sites. We don't think we need to activate every single receptor in the family to turn on the vegetative stress response pathway, and we now have a chemical tool for activating ABA signaling synthetically, and we now have new tools for using constitutively active receptors. And with that, I'll thank the people who did the work. Sung-Yul Park is a research associate in my lab who did the large part of the first half of the experiment, identifying the receptors in the yeast-2 hybrid and characterizing them biochemically. Soph Muskuna did the constitutively active receptor work, and Masanori Okamoto identified quinabactin and did all the experiments I showed on that. These are my collaborators involved in the structural work who I acknowledged already, and have been very generous to receive support from NSF and Syngenta. And with that, I'm happy to take questions.